yet important group of scientists who study virus in the air. Her research team adopts an interdisciplinary approach, integrating studies in physics, chemistry, and biology to characterize emission, transportation, transformation, and fate of contaminants in the air. Professor MARR received a BS in the engineering sciences from Harvard College and a PhD in civil and environmental engineering from the University of California at Berkeley. And she completed her postdoctoral training in Earth, atmospheric and planetary sciences at the Massachusetts Institute of Technology. Professor MARR has worked with the U.S. Centers for Disease Control and U.S. National Academies of Sciences, Engineering, and Medicine to advance the conventional knowledge about virus transmission and recommended guidelines for protecting public health. She's been interviewed hundreds of times and contribute op-eds to the New York Times and the Washington Post about transmission of COVID-19 to raise public awareness of COVID-19 over the past 2 years. Professor MARR has published over 140 papers and is a fellow of the American Association of Aerosol Research and of the International Society of Indoor Air Quality and Climate. So we cannot think of a more qualified person to deliver the following lecture entitled Aerosol Science Transmission of COVID-19 and Other Respiratory Infections. So without further ado, please welcome Professor MARR.
Good morning. It's a real pleasure to be giving a plenary at the Asian Aerosol Conference. I want to thank.
So I want to make sure everybody can see the video and it sounds clearly now?
Yes.
Yes?
Yes,
Okay.
yes, it does. Yeah, it does, at least for me. Thank you.
Okay, thanks.
The conference organizers for inviting me. I wish I could be there with you in person in Taiwan. Unfortunately, due to continuing travel restrictions, I was not able to come. But I was there in 2017 to 2018 on sabbatical at the College of Public Health at National Taiwan University. And my family and I really enjoyed our stay there. We love the land and the culture and the people, and we look forward to returning as soon as we can. I'd like to start out by acknowledging the students, postdocs, and other faculty collaborators who've worked with me on this research over the years, and also the funding agencies that have supported this research. I'm going to address two main questions in this talk today. The first is, why has there been so much controversy over COVID-19 transmission routes? That will be briefer. And then secondly, we will delve into microdroplet chemistry or aerosol chemistry to see whether that helps explain the seasonality of respiratory viruses. This is a question that my research group has been exploring for the past roughly 13 years or so. When the pandemic began, the New York Times published this article originally in January. How bad will the coronavirus outbreak get? Here are 6 key factors. One of the factors was how far viruses travel. And based on conventional wisdom, according to the medical community, they said that coronaviruses can travel only about 6 feet from the infected person. It's unknown how long they live on surfaces. On the other hand, some other viruses like measles can travel up to 100 feet and stay alive on surfaces for hours. In response, I tweeted out, not sure I believe that coronavirus droplets fall to the ground within a few feet, while measles, chickenpox, and tuberculosis can travel about 100 feet. But why do people think this? It was a bit of a rhetorical question because I've been immersed in this field long enough to know that some of the traditional thinking about transmission and it being based on large droplets was erroneous, probably. So traditionally, people have thought that respiratory viruses, such as those that cause colds and the flu, are transmitted by large droplets that are released when someone coughs or sneezes directly into someone else's face, and those large droplets could be carrying virus. They would land in the other person's eyes, nostrils, or mouth, and then cause infection. I think what people did not recognize is that when people cough and sneeze, and in fact, when they talk, they are releasing hundreds of tiny droplets, aerosol particles, for every large droplet that is released. And these are most concentrated close to the person who released them, and they can spread throughout the room and accumulate over time if the ventilation is poor. And so people at a distance can still breathe these in, although people who are closest are more likely to be exposed. And there was some nice work by Dr. Hugo Lee showing that at close range, your exposure is dominated by inhalation of the aerosol particles rather than the spray of the large droplets, unless you're really uncomfortably close. So what got us first thinking about this? Well, in 2011, we published a paper where we had looked at collected air samples in a student health center, children's daycare center, and on airplanes. And we size fractionated the aerosols using a Ceutis cascade impactor. And we analyzed each stage for influenza virus. We found that over half of the virus, in terms of its RNA gene copies, was associated with particles smaller than 2.5 microns. These are fine particles that can remain suspended in air for many hours, if not longer. So why was droplet transmission the default when it came to transmission? Well, we have to go back to the 1800s and earlier, when miasma theory dominated thinking about how diseases were transmitted. Miasma is Latin for bad air, and the thinking was that it could be floating around in the air, especially associated with dead and decaying bodies, that these vapors would come off those areas and could waft through the streets. People could breathe them in and become sick. Germ theory, of course, came about during the 1800s. And then there was a highly influential book published by Charles Chapin, a public health leader, called The Sources and Modes of Infection. And he wrote that instead of kind of in reaction or in pushing back against miasma theory, he wrote that contact infection affords a better explanation, and that ideas in regard to airborne diseases are entirely erroneous. He said they were sprayborne for only 2 or 3 feet. This idea really persisted until the current pandemic. In 1934, William and Mildred Wells published a paper in which they really defined droplet versus airborne transmission. They said that droplet infection was transmitted by things that are rapidly removed from the air by gravity within a short distance from the source, and that airborne infection was caused by droplet nuclei, which was thought to be the dried residue of larger droplets that were suspended for long times and long distances. This distinction pivoted around a size of about 100 microns, or 0.1 to 0.15 millimeters. And they based that on an analysis of the time that it would take for droplets to evaporate versus the time that it would take for them to fall to the ground. So for those smaller than about 100 microns, they would evaporate in a shorter, before they falling to the ground, so they could remain suspended, but the larger ones would fall to the ground fairly rapidly. And then in 2007, the US Centers for Disease Control wrote in one of its guidelines that droplets traditionally have been defined as being larger than 5 microns in size. And droplet nuclei, or particles arising from the desiccation of suspended droplets, have been associated with airborne transmission and defined as less than 5 microns in size. And because of this, there was this mental model that transmission occurred, again, mainly by these large visible droplets, and that droplet nuclei or aerosol particles were really only relevant from so-called aerosol generating procedures, because I think there was a lack of recognition that when people talk and cough, they're also releasing a lot of aerosol particles that can carry virus or other pathogens. Well, how did this error come about? Because in 1934, Wells stated that the cutoff should be around 100 microns, and yet by 2007, that had become 5 microns. We worked together with a collaborator of mine in the history department at Virginia Tech, and he brought on board a graduate student in medical rhetoric. And she did a lot of kind of backward searches through the literature and found that what likely happened is that people conflated how droplets and aerosols move through the air with where they deposit in the respiratory tract. So when someone talks, they release droplets and aerosols of a wide range of sizes. The droplets, in terms of if we wanted to find those that travel ballistically through the air and fall to the ground within maybe 3 to 6 feet or 1 to 2 meters, those would be larger than 100 microns. The aerosols would be smaller than 100 microns. And those are small enough to be inhaled also. If we look at deposition, though, of particles in the respiratory tract, it is things that are larger than 5 microns that would deposit in the upper respiratory tract, and they would have to be smaller than 5 microns in order to reach the lower respiratory tract. And somehow along the way, probably due to the dominance of tuberculosis in thinking about airborne transmission, with the bacterium that causes tuberculosis, it can only initiate infection deeper in the respiratory tract because that's where the receptors are. But somehow this 5 micron cutoff for deposition in the respiratory tract became essentially kind of the cutoff for how things move through the air. And so you can see how instead of 100 microns, the cutoff became 5 microns. And thus it seems that aerosol transmission somehow became diminished. There's a dramatic telling of this story in this Wired article, the 60-year-old scientific screw-up that helped COVID kill. In August of 2020, several of us participated in a workshop on airborne transmission of SARS-CoV-2 led by the US National Academies of Sciences, Engineering, and Medicine. And I propose that we should be defining transmission by the exposure pathway instead of by necessarily by size or of the droplets. And so really large droplets are sprayed onto the body and they impact onto our eyes, nostrils, or mouth, or perhaps they land on a surface where they could contaminate the surface. Someone could touch it and transfer the virus into their body. On the other hand, aerosols are things that can be inhaled into the respiratory system. This distinction is important because it drives control strategies. Face shields will work against large droplets, but not against inhalation of aerosols. And distancing will also work against large droplets, but not against inhalation of aerosols. This distinction also drives the infectious dose. There are studies showing that it takes fewer of the viruses to cause infection if they are inhaled deeper into the respiratory tract than if they are just dripped into the noses of the new hosts. And it also may drive the severity of disease. And the physics-based cutoff for what we would then talk about in terms of spray versus inhalation is between 60 to 100 microns, depending on the velocity of the exhaled jet and ambient environmental conditions, namely humidity and other airflows. So now let's look at this question of what drives seasonality of some respiratory infections. This shows the incidence of flu in different parts of the world. And in temperate regions, such as Bismarck, North Dakota in the US, shown in the upper left-hand corner, and Sydney, Australia, shown in the lower right-hand corner, there's a wintertime peak, a very clear wintertime peak in flu incidence. So in Bismarck, we see it around December, January, February, and then tapering off in toward the spring. In Sydney, Australia, we see a peak in kind of June, July, August, September. On the other hand, in tropical regions, such as Singapore, shown in the upper right, and Fortaleza, Brazil, shown in the lower left, the incidence follows a different pattern. In Singapore, there's actually 2 peaks, one in the middle of the year and one at the beginning or end of the year. In Fortaleza, Brazil, it's kind of in the March, April, May period. So why is this? Well, there are many factors that contribute to the transmission and incidence of disease. These include factors regarding the host, their immune defenses, and factors regarding the environment. The outdoor environment affects the indoor environment. We think most transmission probably occurs indoors, so it's really those variables that matter indoors. There's characteristics of the virus and droplet that matter, which is what I will focus on. And then shown at the bottom, human behavior, of course, affects contact rates, and there's other interventions, hygiene that matters. So I'll be focusing here on the virus and droplet characteristics, like the viability of the virus, droplet dynamics, and droplet size and matrix, and how those might contribute to seasonality. I'm going to review characteristics of viruses in the air. First of all, size is critical. The airborne virus is not naked. SARS-CoV-2 is about 0.1 microns in size and diameter, similar to the flu virus, but it is carried in respiratory fluid droplets. That's how it's released from our bodies. And these droplets may range in size anywhere from 0.2 to 100 microns or more in diameter. The droplet shown here to scale is 0.5 microns. And it's the size of this carrier droplet or aerosol that defines its transport. And we know that the settling velocity can be readily calculated from the droplet size and the density of the components there. And the size is really important because it determines how long the droplet can stay aloft, how far it can travel, how quickly it falls to surfaces, where it deposits in the respiratory system, and how efficiently it is removed by masks and filters. The physics is the same for all viruses. Really, what's important is the carrier. There may be differences between viruses in terms of how well they survive while they're traveling in the droplets, how many are released from the infected host, and how many it takes to become infected, to initiate infected in a new host. And for reference, a 5 micron particle, if we go back to that cutoff that was thought to differentiate large droplets from aerosol particles, well, a 5 micron particle takes over 30 minutes to settle to the ground from a height of 1.5 meters. So it's clearly not something that travels in a ballistic pathway and that falls to the ground quickly within a close distance. The respiratory fluid droplets are not just water. They consist of other components, just like our sweat has salts in it. Our respiratory fluid has salts and proteins and other things in there. The non-volatile solutes make up anywhere from 1 to 9% of the initial mass of the droplet. And that consists of inorganic components such as sodium chloride, potassium, and also organic material in the form of proteins. There's some surfactant present. And then the virus itself is likely to make up less than 1/1,000,000th of the mass of those non-volatile solutes. The image on the right shows a electron microscope image of respiratory droplets. They're about 1 micron in diameter. You can see the outer ring is kind of the evaporated residue after they had evaporated on the substrate. And then you can see large crystals in the middle. And by EDX, those were shown to contain potassium, calcium, chloride, carbon, and sulfur, exactly what you would expect for kind of salts and proteins being the main components of this fluid, non-volatile components. So the seasonality, I'm going to focus on temperate regions where we have that wintertime peak. Well, is it because there's more evaporation at lower relative humidity? In temperate regions, we heat the air in the wintertime, and so it becomes very dry. The relative humidity is very low, typically between 10 and 20%. Well, under those conditions, the droplets would evaporate more, they would be smaller, they could stay suspended in the air more. So we developed a model to predict virus dynamics in indoor air in a well-mixed, we assumed a well-mixed environment where someone would release droplets of all sizes containing virus into this space, and they would be removed by ventilation, gravitational settling, and then inactivation, which is a process whereby the virus loses its ability to infect cells. It's somehow biochemically disabled or damaged. And so we were able to develop a mathematical model based on mass balance, where we can predict the concentration of infectious virus in aerosols of diameter D. So we did this size resolved with first order removal by these 3 processes. The settling velocity, V sub T, depends on diameter, as I mentioned earlier. the diameter depends on relative humidity, according to the Kohler equation, which I'll show in the next slide. And the inactivation rate K also depends on relative humidity, according to many studies in the literature. And so built into this equation is a dependence on relative humidity. The Kohler equation is shown here, and this graph shows the ratio of the final to the initial diameter of a droplet that is released and equilibrates with ambient humidity. So when they leave our respiratory tract, they are coming out of an environment that's saturated at around 100% relative humidity. And so if we have a droplet that's 10 microns in size, contains salts, proteins, maybe our virus, it then hits ambient air, and the relative humidity is shown along the x-axis, and because the air is drier, that droplet is going to evaporate until it reaches equilibrium, where the vapor pressure above the surface of the droplet is the same as that of the surrounding environment. So if the ambient air is about 20% relative humidity, that droplet will shrink to about 4 microns in size. The exact size depends on the chemical composition of the respiratory fluid droplet. But you can see that the solutes in there become much more concentrated as the water is lost. And even if the relative humidity is 80%, that droplet still evaporates a lot, down to 40 to 50% of its original diameter. And so you can see that at all but really high humidities, this droplet is going to shrink rapidly in size, and the chemical composition, the chemical microenvironment surrounding that virus particle is really going to change. And this assumes that the droplets reach equilibrium. Some of our studies suggest that there's that maybe a kind of a shell, organic shell forms on these droplets as they're evaporating that would inhibit further evaporation. But anyway, this shows some of our model results. It shows the concentration of infectious virus as a function of size. And we're going to start out at 0 minutes. So this is the person coughs. We assume those particles, droplets, are instantaneously well mixed throughout the space. You can see that there's a peak in the size distribution, somewhere around 40 to 50 microns in size. So that's where most of our viruses are. But the largest ones rapidly settle out. And so within a minute, we've lost the largest ones. And then the whole curve shifts to the left, because everything shrinks, right, to about less than half its original diameter. And as we proceed further in time, we continue to lose the larger droplets due to gravitational settling. And eventually we end up with a size distribution where the peak in terms of numbers of infectious viruses is less than 10 microns. So again, there's a size shift due to immediate evaporation and gradual loss of larger droplets by gravitational settling. Now, if we compare the different removal mechanisms and their efficiency, the left-hand graph shows that at an air change rate of 1 per hour at different humidities from 10 to 90%. And you can see that gravitational settling is very efficient for removing virus particles. We assume that they are initially homogeneously distributed among the droplets by volume. And at higher relative humidities, 90% shown in purple, we have a somewhat higher removal efficiency because those droplets stay a little bit larger and settle out faster. Ventilation is not very efficient at this air change rate, and it's independent of relative humidity. Inactivation, though, does depend quite a bit on humidity. You can see that it's highest at 90% relative humidity and 0 really at 10% relative humidity are dry conditions. And then we can also look at this under a much higher ventilation rate of 10 air changes per hour. The only difference here is that the removal efficiency by ventilation is now much higher than it was before. So we saw that in terms of removal efficiency, relative humidity does affect removal by settling, but it has a bigger effect on virus inactivation. And so that led us to further studies of how does virus viability vary with relative humidity. Now, I didn't show this earlier, but I mentioned that inactivation depends on relative humidity. And we pulled the relationship from this study from the 1960s by Harper on flu virus, where he looked at he injected influenza virus into a chamber and pulled it out at different times to look at how much had decayed at different humidities. And at low relative humidity, the decay rate was fairly small. Here, the y-axis shows decay rate. It's 0 towards the top and higher going down. That's because in all of these graphs subsequently, I'm going to show higher viability or better survival upward toward the top on the y-axis. And so at low relative humidity, the decay rate is small. And as we go to higher relative humidity, the decay rate increases. There was a nice linear relationship here. That, however, was very different from other results that we found in the literature, which suggested more of a U-shaped relationship between virus viability and relative humidity. So in these other studies, we saw that the decay rate was highest, largest, so that's the dip in the U, at somewhere in the mid-range humidities. And the difference was we think was due to the composition of the media, the liquid used to aerosolize the virus. And with the monotonic relationship that we used in our model, that was shown mainly in salt-containing solutions, whereas the U-shaped relationship was seen in solutions that contained also protein. And so we started to conduct some of these studies ourselves, and we looked at virus viability in droplets sitting on surfaces and also in aerosols. So for the droplets, we set up a chamber with controlled relative humidity using different saturated salt solutions. And then we built a custom rotating drum shown in the lower photo to keep aerosols suspended for long enough that we could, for an hour or more, so that we could look at how viability changed over that time period. We exposed the viruses over a wide range of relative humidities from 20 to 99% for 1 to 3 hours, and we varied the fluid composition also. Here are some of the results that we found. The top figures show survival of flu virus in droplets. The y-axis is viability in terms of percent, so 100% means that all the virus survived. This was after, I believe, 3 hours sitting in droplets on plastic surfaces. And in both these figures, you can see that V-shaped relationship or U-shaped relationship. On the left-hand side, the PBS and DMEM refer to culture media that we grow the virus in or suspended in that consist mainly of salts and very little protein. And you can see there's a big dip in viability in the mid-range humidities around 50 to 70-ish%. On the right-hand side, we supplemented those media with FCS, fetal calf serum. And so the viability was better. We don't see that huge dip at the mid-range humidities, but there was still a slight U-shaped relationship. And we've done this also with flu virus and aerosol, suspended aerosol particles, and also with some model viruses, bacteriophages, which are viruses that infect bacteria rather than other animal cells. And we used ones called Phi6, which is enveloped, has a lipid envelope similar to flu virus and SARS-CoV-2 and is similar in size, and also MS2, which is non-enveloped and is a bit smaller. But you can still see the relative viability here shown on the y-axis. We have that dip in humidity where we have this U-shaped relationship, and somewhere, depending on the virus, there's a dip somewhere around kind of 40 to 80% relative humidity. This U-shaped relationship was also seen with SARS-CoV-2 in droplets in data experiments conducted by Vincent Munster's group at Rocky Mountain Laboratory. And the different colors there on the lower right are showing different temperatures. But at any temperature, again, you see this dip in viability at around 65% relative humidity. So why is there a U-shaped relationship between virus viability and relative humidity? The virus itself, again, is not naked, and so it's not directly exposed to ambient humidity, but really it's the humidity controls evaporation, which then controls the chemical microenvironment surrounding the virus. So we have been looking at evaporating droplets. We start out with 1 microliter droplets that are pipetted onto surfaces and looked at how fast they evaporate at different humidities. This figure shows the normalized weight over time, over an hour. at different humidities. And you can see that at high relative humidity, 98%, we have very slow evaporation of the droplets. They go from their initial weight to about 80%. So they lose about 20% of their water over that hour. And then at very low relative humidities, we have rapid evaporation. They evaporate within 20 to 30 minutes at humidities of 23%, 33%, and 43%. And then in the mid-range, we have this slower evaporation. This corresponds to a change in solute concentrations, which we calculated based on the loss of water. So if the droplet shrank such that it lost half its water, we would expect the solutes to increase in concentration by a factor of 2. And we call that the enrichment factor. Here, we can see that for the very high relative humidities over time, there's very little evaporation. And so there's not much enrichment at 98% relative humidity and at 85%. However, at low relative humidities, we rapidly achieve complete evaporation. And so we reach enrichment factors of 15 or so at those lowest humidities. And then after that, the droplets are completely dried out. And so there's the concentration is undefined and those curves just end. However, at these medium relative humidities of 43 and 55%, we have slower evaporation that isn't complete. So the droplets stay a little bit wet, but they have been evaporating and things have become more concentrated. You can see we reach high enrichment factors greater than 40, a factor of 40 in both these cases at 43% and 55% relative humidity, the red and the green. Well, what does this mean for the concentrations of the solutes? And here we show over time the sodium chloride concentration in grams per liter. And the scale goes from green, 0, up to red, 350, or really 360, which is the saturation concentration. Above that, we expect the sodium chloride to crystallize out of solution. And you can see that at very low relative humidities on top, at 23%, we rapidly reach that really high concentration, and then the droplets have dried out, they've effloresced, everything has crystallized, and so the concentration is no longer relevant. And at the very high relative humidity shown at the bottom of this graph, we stay in the green or maybe the yellow range because there's very slow evaporation, the sodium chloride concentration stays near its initial value in these droplets. But really it's at this mid-range humidity at 55% where we reach very high concentrations shown in red around half an hour, 30 minutes, and then we stay there because the evaporation continues slowly, but the droplet has not effloresced. And in that case, the virus is exposed to high concentrations of solutes for a long time at these mid-range relative humidities. Another factor is the physical characteristics of the droplets. This is showing work done in collaboration with Brian Davis. And this shows in flu virus viability in both DMEM, one of our culture medium, and in human mucus as a function of relative humidity. And the blue color, the dark blue color, indicates that the droplets are still liquid-like. So you can see in that range, if we go from 100% towards the left, we see decreasing viability as the humidity drops. So things are becoming more concentrated. But then once we hit that light blue region, which is where the droplets have reached this amorphous or semi-solid phase, we see that the viability actually is higher than it was right at that transition point before the droplets became semi-solid. And then at very dry conditions in the gray area, the droplets are partially crystalline and the viability is maintained. Viruses seem to survive quite well when they're fully desiccated. And so we think that this semi-solid phase in the light blue at mid-range relative humidity may actually inhibit diffusion of the reactants and slow decay. Now, what is the actual mechanism of virus inactivation? We don't know yet, but we have been trying to, we've been manipulating our droplet composition, one component at a time. We've done this with salt, sodium chloride. So the graph shows relative viability or survival of viruses at 3 different humidities and 3 different sodium chloride concentrations, 0 shown in black, 1 gram per liter shown in red, and then 35, which is like seawater, shown in blue. And you can see that viability in general seems to decrease as relative humidity increases. And then there's an inconsistent effect of sodium chloride. In some cases, when we go from 0 to 1, we have a decrease or an increase. And when we go from 0 to 35, that again, it's unclear. With pH, there is more of a clear result. Again, this is at 3 different relative humidities, and we have pHs of 4, 7, and 10. So red is the pH of 7, which is neutral, and so we have pretty good virus survival at that condition. And then at 4, we were not able to detect any infectious virus. So 4 acidic conditions definitely seem to be harmful for the virus. And at a pH of 10, we saw reduced viability also. We've done this also for protein in the form of bovine serum albumin, and that seemed to be protective at 50% and 80% relative humidity. And then we looked at surfactant in the form of SDS, and the effect of that was inconsistent. So what exactly is the culprit here? We've talked about salt and pH, protein, and surfactant. Another possibility is reactive oxygen species. There's been some nice work done by Richard Zare showing spontaneous generation of hydrogen peroxide in aqueous microdroplets. And he also found that the amount of hydrogen peroxide increased with relative humidity up to 50% over this range that they looked at. Another factor is spatial heterogeneity in pH within droplets. Together with my collaborator, Peter Vickersland at Virginia Tech, we used gold nanoparticles that were as pH sensors, they were coupled with a pH sensitive dye. And we put these into a solution, aerosolized it into the microdroplet sitting on a surface, used Raman spectroscopy then to estimate the pH at different locations within the droplet. And the graph shows pH as a function of distance from the droplet center. Right at the center at 0, we saw a pH of 11. The pH of this solution that we started with, this phosphate buffer, was 7. And so the middle of the droplet was quite a bit more basic. And then as we moved away from the center towards the edges on either side, we saw that the pH was lower. Other possible culprits are carbon dioxide, ammonia, nitric acid, and other atmospheric oxidants that could get into the droplet. So this is a question we are still looking into along with other groups around the world. Well, how does this all relate to seasonality? To come back to my original question. The seasonality, again, of influenza is well documented. And there was a recent publication showing that there's a U-shaped relationship between the reproduction number, the effective reproduction number, R sub T for flu, and absolute humidity measured outdoors. And so you can see in that left-hand figure that the predicted effective reproduction number, R sub T, is higher at the extremes, at very low absolute humidity and very high absolute humidity. Well, again, transmission takes place mostly indoors, probably, and we believe that absolute humidity here is serving as a proxy for indoor relative humidity. It's true that absolute humidity is pretty well correlated with indoor absolute humidity, and because the temperature indoors usually falls within a fairly narrow range, that relationship also holds true for indoor relative humidity, shown here on the right-hand side with some work that we published recently. So we do see a U-shaped relationship in flu transmission, U-shaped with humidity. Well, how might humidity control transmission? Let's start over on the right-hand side here. They're both physics and chemistry that factor in here. At very high relative humidities, so the droplets come out of our respiratory tracts. They were present at 100% relative humidity. Well, at very high relative humidity, a droplet that comes out that was 25 microns in size is going to stay about that size, and it'll settle rapidly in about 1 minute. And in terms of the chemistry, the droplet is going to remain, the virus will remain at close to physiological conditions in the fluid that it was released from without much further modification. At medium relative humidities, that droplet is going to evaporate down to about 40% of its original size, so around 10 microns, and that will take 8 minutes to settle. So it's going to be in the air longer. And in terms of the chemistry, that droplet evaporates a lot, but not completely. And so we have this very highly concentrated solutes in the droplet that could be harmful to the virus. At low relative humidities, that droplet again can stay suspended longer because it is now 10 microns instead of 25 microns. And the droplet has effloresced completely, and then we know that there's minimal decay under those conditions. So in terms of the seasonality, just kind of putting this all together, at low relative humidities in the temperate regions, it's below 50%, really well below 40% usually. We have wintertime indoor air. We have a combination of smaller aerosols and minimal decay of virus. And maybe that is something that contributes to our flu season. At medium relative humidities, such as we see in the spring, summer, and fall, indoors at least, we have small aerosols, and we also have faster decay of the virus. And at very high relative humidities, like in the rainy season in tropical regions, there's less decay and perhaps there's more contact transmission from large droplets that have settled. That's a hypothesis that's been floated, but we don't have good confirmation of that yet. And so to conclude, there are dynamic processes that control the fate of viruses in the air and influence the efficiency of airborne transmission. And I've also shown that humidity affects virus survival by modulating the microenvironment in droplets and aerosols. This very much depends on the chemical composition of the fluid. And we've recently shown with collaborator Seema Lakdawala, an influenza virologist, that when we use more physiologically realistic fluids containing mucus from human cells, that we don't see that same sensitivity to humidity. So it's still really an open question. And then the figure here at the bottom is from a paper I co-authored with several other people in Science on airborne transmission of respiratory viruses, if you are interested in learning more about them. Thank you very much for your attention.
Well, thank you, Professor MARR. Since Professor MARR is not available online with us for the moment, I guess as a moderator for the talk, I'll simply take the liberty of closing this presentation by thanking her again, but also addressing that it has been a rather comprehensive and very, very well-structured talk. Beginning by a very, very sort of able information historically, and giving us an overview as to how all these different aspects of factors and sort of inferencing information has come about, and where in the beginning of the pandemic certainly has triggered and motivated a lot of debates and concern. But scientifically, we have came a long way by putting together constructed, well-constructed sort of a dynamic picture of analyzing all these available informations. So I think in the end, Professor MARR emphasized again how in the future there requires and warrants a lot more collective efforts in putting together the understanding toward how we would view the different aspects of inferences from either an aerosol standpoint or sort of a droplet standpoint in the preventing and controlling of the future pandemic. So with that, I want to thank the organizer for the opportunity, and again, thank Professor MARR for her work and her presentation. So with that, I'm going to relay and give the floor to the next moderator, which I believe Professor Wu is ready.